This video will explain how fluid movements along the cochlea affect the organ of Corti. It will show how sound waves of differing frequencies travel along and are detected at different points of the cochlea. It will list the functions of the inner and the outer hair cells. It will explain how the inner ear can detect tiny differences in the frequency of a sound and describe how the inner ear responds to sound. It also looks at the hearing process after sound reach the inner ear and the cochlea. It describes how the movements of the oval window are converted into electrical signals that are perceived as sounds. The system is incredibly sensitive to small changes in frequency and intensity, and this session will discuss how such sensitivity is achieved. The fluids of the inner ear are non-compressible. Therefore, the movement of the stapes in the oval window transfers directly to the perilymph in the cochlea. It generates a pressure vibration, or wave, which travels around the cochlea and causes a movement of the round window. The movement of the round window is in the opposite direction to that of the oval window. So if you look at the arrow, when the oval window is moving to the right, the round window is moving to the left. Sound vibrations in the scala vestibuli are transferred into the scala media, which contains endolymph, by movements of Reissner's membrane. As the wave passes the helicotrema and continues along the scala tympani, the basilar membrane is also displaced in a similar way. The basilar membrane reacts more to vibrations of the inner ear than most of the other structures. The organ of Corti resides on this membrane, so these vibrations are readily transmitted to it. As the basilar membrane is not of a uniform shape and stiffness, areas along it show greater displacement for some frequencies than for others. Low frequency or low wavelength tones cause maximum displacement at the apex of the cochlea. High frequency tones, which have a short wavelength, cause maximum displacement at the base of the cochlea. As the basilar membrane moves, the outer hair cells are twisted. The stereocilia, which are on the tips of the outer hair cells and embedded in the tectorial membrane, are moved. This twisting or shearing motion bends the stereocilia towards the tallest stereocilium and then back in the opposite direction. Although the stereocilia on the inner hair cells are not thought to be connected to the tectorial membrane, they also experience this shearing effect if the stimulus is powerful enough at that point along the basilar membrane. This causes them to make contact with the tectorial membrane. Tiny transduction or tip links join the tips of the stereocilia. At rest, the inner and outer hair cell has an electrical charge of approximately negative 60 millivolts and the endolymph around it of positive 80 millivolts. As the transduction links tighten, they allow positive ions to enter the hair cell, which in turn trigger the release of neurotransmitters at the base of the cell. These neurotransmitters fire a wave of depolarization, or a nerve impulse, down the afferent nerves. This is referred to as an action potential. The hair cell then returns to its resting state. If the stimulus is a loud sound, the afferent nerves will fire more quickly than for quiet sounds. However, there is a maximum rate of firing that can be achieved, as all cells need time to recover or repolarize. Repolarization is the return of the cell to its resting state. Most everyday sounds are quite complex and made up of many different frequencies. The human ear can detect sounds in the frequency range of 20 Hz to 20,000 Hz or 20 kHz. The human ear is also able to detect tiny changes in frequency or pitch of as little as 1 Hz and intensity changes of less than 1 decibel. The travelling wave theory alone does not adequately explain how our hearing can be so sensitive. As we have seen, different frequencies move different parts of the basilar membrane. It is thought that the outer hair cells in that region are then stimulated by the efferent neurons innervating them. This causes them to contract. Outer hair cell stereocilia are embedded in the tectorial membrane. This contraction therefore drags the basilar membrane upwards. Since the inner hair cells also sit in the basilar membrane, they are moved closer to the tectorial membrane in the stimulated region. The inner hair cells are now closer to the tectorial membrane, and their stereocilia are more likely to touch it and generate an action potential. Contraction of the outer hair cells has therefore increased the sensitivity of the inner cells in that region. It is the inner hair cells that are mainly responsible for responding to and detecting sounds. The majority of neurons innervating them are afferent and carry impulses towards the brain. The outer hair cells do respond to stimulation, but their main function is to contract in response to stimulation, drawing the basilar membrane upwards and effectively increasing sensitivity in regions of activity. This process requires energy and is therefore referred to as an active process. This fine-tuning explains why the ear is able to detect such small changes in intensity and frequency. Sound waves travel along the cochlea. The position of maximum displacement is dependent on the frequency of sound. High frequencies cause most displacement at the base of the cochlea and low frequencies at the apex. Movement of perilymph generates vibrations along the basilar membrane, causing the inner hair cells to be activated as their stereocilia begin to touch the tectorial membrane. Outer hair cells, close to areas of activity, contract, drawing the basilar membrane upwards and therefore increasing the sensitivity of the inner hair cells in that region. This process explains why we can detect very small changes in frequency and intensity.